Hi, I'm Marshall, and I'm going to be talking about the process of muscle contractions. So, the first part to the process of a muscle contraction is the stimulus, and this is an event in the real world that makes our body react. And during the PowerPoint, I'll be using the example of a rugby ball coming towards you as the stimulus for a muscle contraction. And what actually happens is the stimulus is received by our senses, and in this example, the sense will be our eyes because we see the ball coming towards us. And the stimulus makes the sensor send a message down sensory neurons, which is sent to the brain. And this message could say anything. For example, it could say, right, catch the ball. And the message would be received by the brain, which then decides, right, what am I going to do regarding the stimulus? And decision could be to try and catch the ball. And so the brain would send another message down motor neurons in our nervous system towards the receptor muscles in our hands, saying, get ready to try and catch the ball that's coming towards you. But the problem with this is that at the end of our nerves, between them and the muscle fibers, there are gaps. And the slightly bigger gap is called a neuromuscular junction. And inside this junction is a smaller gap called a synapse. And this synapse is actually what stops the impulse being easily received by the muscle fibers. So in order for the impulse to get over the synapse, it has to travel in the form of a chemical. And then once that impulse has made it across to the fiber knows, okay, I've got to contract. Our muscle fibers are made up of many myofibrils, which contain lots of sections called sarcomeres. And these sarcomeres are adjacent to each other and have two filaments. As we can see in the picture on the screen, they have a thinner filament called actin and a thicker filament called myosin. And these alternate across the whole of the sarcomere. And this is why if we look at skeletal muscle tissue under a microscope, we can actually see that it looks striated or striped. So the muscle actually contracts when the filaments slide past each other, and in order for this to happen, the myosin has to gain energy so that its arms can pull the actin along. This energy comes from a substance called adenosine triphosphate, which we'll call ATP. So the myosin arms are ready now that it's gained the energy from the ATP, but the problem is the actin isn't ready. And this is because the actin has binding sites on it, which is where the myosin arms need to connect to. And these binding sites are actually covered by two proteins. As we can see on the screen, they have a tropomyosin protein, which is a string-like thinner protein, which is partnered by troponin, which is a smaller round protein. And in order for the muscle to contract, we have to move these two proteins so that the myosin arms can connect to the actin binding sites. But the question is, how do we move them? And the answer to this is we actually introduce calcium, and calcium is stored in the sarcoplasmic reticulum. And what we can do is we can react the calcium with these two proteins, and what it does is it makes them change shape and ultimately moves them away from the actin binding sites. So the two proteins are now ready. The actin binding sites are not covered, and the myosin arms have got the energy to connect. And so what happens is the myosin arms form a cross bridge, as we can see in the picture. They reach across and they grab onto the actin binding sites. And what they'll do is they'll perform something called a power stroke. And the power stroke is basically the process of myosin pulling the actin alongside it. And because of the actin being attached or anchored to the ends of the sarcomere, which we will call the Z line, it actually means that these Z lines are being pulled towards the middle of the sarcomere, which is called the M line, and this results in the sarcomere shortening in length. Due to the myosin arms having to perform potentially many of these power strokes, they have to be constantly supplied with energy. And this means that every time the arms perform a stroke, more ATP is supplied to them. And this results in the ATP becoming ADP, which is adenosine diphosphate. And this leaves one single phosphorus molecule on its own. So that's half of a muscle contraction, but the other half of it is how do we stop the muscles from contracting? And what we actually do is, let's say that the ball is now caught and we don't need our muscles to contract anymore. What happens is our brain will stop sending the impulse or message saying catch the ball down to the muscles, meaning that they do not know what to do now, and they voluntarily stop contracting. And this is actually helped by stopping calcium being released in order to move the troponin and tropomyosin away from the binding sites. So in conclusion, if there's no calcium, it means that the proteins can come back and cover the actin binding sites, meaning that the myosin arms cannot attach to them and ultimately contract the muscle. So that was me talking about the process of muscle contractions. Thanks for watching.